All right. This is TWIS, This Week in Science, episode number 491, recorded on Thursday, November 26th, 2014. This is real. Hey everyone, I'm Dr. Kiki, and you know this is real because this is This Week in Science, and today we are going to fill your heads with a bit of spacefaring DNA, frog maps, and your imagination. But first...
Disclaimer, disclaimer, disclaimer. We gather together here at the table of science to give thanks. Thanks to the science-minded pioneers of yesteryear who forged for us a world of reason so that our minds might plant and harvest in fields that produce rational results and make real progress in our standard of living. Thanks to the researchers of today who provide a weekly bounty of new knowledge, insights, and awe-inspiring discoveries. Thanks to the engineers of technology who have made production of and access to the show simple and readily available to all who seek it. And above all else, thanks to you, the listening audience, the minions of TWIS, without which the show's hosts would simply fall into a new category of the Diagnostic Statistical Manual of Mental Disorders, identifiable as speaking to oneself aloud about science news without an audience. The tradition of giving thanks is a good one that truly deserves to be celebrated. And I can't help but think that this is what we are doing each time we say, This Week in Science! Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to Kirsten and Blair.
Good science to you, Justin and Blair, and everyone out there in science land, which is really the world, because that science is all about the world and the universe. Hey, everybody, it's This Week in Science. We have a great show ahead, right? We're ready to give thanks. I'm giving thanks every minute of this show. Thank you for listening, because it is Thanksgiving Eve, or as Blair would like to have it, hashtag TWISgiving.
Hashtag TWISgiving, y'all.
Right. Let's hashtag it. Let's make it ours. Oh my goodness. On this week's show, we've got a bunch of science news. I've got news stories about how brains work, tough space DNA, and a few gut bacteria. What do you have, Justin?
I've got why fighting with family is a good thing, poo rockets, and cheap ambulances.
Poo rockets. Really?
Really?
Okay. And Blair?
Ooh, it's an all cold-blooded twist for me today. I have frogs, turtles, and fish.
Ooh.
And maybe a peacock spider. We'll see.
Which technically are have blood, kind of, but yeah, kind of, sort of. Yeah. All right, let's dig into this fun soap opera I'd like to revisit in my first story. A story from last week in which we talked about a study. Excuse me, I had to sneeze. A study regarding the use of Triclosan. And in this study, researchers published in the Proclamations of the National Academy of Sciences, a study entitled "The Commonly Used Antimicrobial Additive Triclosan is a Liver Tumor Promoter." And the news release that went along with it also promoted the idea that it was a tumor promoter, that it caused cancer in mice. And the study that I was able to take a look at did show that that Triclosan, as we have talked about it many times before, is used in wide use in products for the consumer spectrum, but it also has been known to be linked to health and environmental issues, although it hasn't ever really been linked causatively to stuff. There's a lot of correlation to the health effects as we know them in humans. And so here in this mouse study, they were able to, they showed that it led to, it was basically a triclosan acted as a molecular factor in promoting tumor growth and fibrogenesis within the tumor, within the livers of mice. And there was a commentary posted about this after some people discussed this online, discussed this study by Hilda Bastian over at Scientific American, who she says that this is a bunch of marketing hype and that this study actually overhypes and is going to get people saying, oh my gosh, my hand soap caused cancer in my loved one who has cancer. And people do like to jump to conclusions in that way, and I hope that we did not promote that kind of thinking last week, but I just want to make it I just want to make a statement that this is one study out of a large number of studies, but there are hopefully more studies that will come as we see more studies showing bioactivity and potential negative health effects in species that we use as model animals for our own human health. Mice don't always get it right, as we talked about last week. Also, a story from Justin discussing the genetic similarity and differences between mice. We do have some differences, and so it could be that this liver cancer is something that is only experienced by mice because of their genetic makeup. It could be that it's just a mouse thing and not a human thing at all. But it's out there and it's added to the body, and I do hope that we didn't add to fear-mongering. But at the same time, in the same way that people argue about climate change, that if you can do something to protect yourself, why not do it? So if you can choose to use products that do not contain this compound, that we do not know exactly what it does, but we do know that it has some negative effects, you can choose not to, or you can choose to ignore the news entirely because there has been no ruling by any overarching health body deciding that triclosan is bad. So far, so good for triclosan. That's why it is still in products.
Right.
So
I think there's a few things going on here. There's, just like anything else, there is no 100% solution to all things, right? So you can't then just use, what we're trying to say is you can't use antibacterial soap on everything and never get sick. You can't, it's something that is powerful, and we want to think about using it before we use it all the time, right? So we just want to be informed before we start putting it in every soap dispenser in our house. That's not to say if you have a newborn that you shouldn't be using it, right? And that you're trying to protect your family when there are situations where maybe someone is sick in the house and you have someone in your family who's susceptible to illnesses and you want to make sure that that bacteria doesn't spread. That's fine. But it's something that we just want to be kind of cognizant of as we move around and use these things. We want to look at all the things that might present itself related to that. So I think it's important to look and say, okay, so this has happened. But I think that also it's important to remember, like we talk about the difference between correlation and causation all the time. Yeah. That was one thing in this article I thought was really interesting, is talking about the difference between a cancer causer and a cancer promoter, right?
Yeah. Exactly.
Those are two different things, and so this might make your likelihood of getting cancer higher, potentially, and that's what we found out last week, but it's not going to give you cancer, and those are two totally different things.
Exactly. Exactly. And so someone in the YouTube comments to our video from last week made the comment that I had committed the natural products fallacy in talking about triclosan as something foreign to us, even though it has been found in concentrations. It's been found in high concentrations in just about everybody, including newborns. It's all over the place. But there are all sorts of other things which it's true, and I'll put it out there that there are things like urea, which can cause all sorts of terrible things if it's in high enough concentrations. That's also found naturally in our body, in our breast milk, in all sorts of things. And so what we need to think about here, it's not what I did not mean to commit this fallacy of natural products. It's not as if I'm saying, oh my God, it's in our bodies, it shouldn't be there. What I'm saying is we need to be learning and getting scientists to figure out what concentrations are safe and which are not. Is there a limit to how much we can have in our bodies? There are all sorts of products that are accepted up to certain quantities. So let's just figure that out, okay?
Yeah, and I think we're on our way. And I think maybe we can get to a point where triclosan is not something that is in our dish soap and our hand soap and this and that. And maybe it's something that, I don't know, could be prescribed like antibiotics, or could be in very specific types of soaps for specific things.
Right.
So something that is in so much of stuff, and maybe, all we're saying is maybe, it shouldn't be in everything.
Right. Maybe, just maybe, it shouldn't be in everything. Yeah. But anyway, it's up to you to make the decision, and I just wanted to put it out there that if you want to read it, there's this great blog over at Scientific American Blogs that puts some interesting stuff out there to take into consideration in relation to this triclosan conversation. So I think that's very important to consider. Second story as we get into the show. Have you ever wanted a Star Trek-like force field that you could just be like, I'm going to protect myself?
I have one.
You do?
Mm-hmm.
Well, we have one around the planet.
Yeah, that's the one.
Oh, okay.
The atmosphere.
Yeah, it's in our atmosphere, exactly. Well, it's even more amazing. The researchers from the University of Colorado at Boulder have discovered this barrier to high-energy electrons about 7,200 miles above the Earth. So we know that there are these Van Allen radiation belts that exist around the planet, and they seem to come and go in their strength with the intensity of space weather. However, these, and they kind of protect us. They grab these energetic particles coming from space and through the solar winds, and they mix in with the magnetic field of our own. They're basically held there by the magnetic field of our planet. But there are these, once high-energy electrons zip into the Van Allen belts, they're caught there, and they should slowly diffuse into the atmosphere of our planet. But they don't, or at least they're not doing it in the way that researchers thought they should. And so upon looking at our atmosphere and our even further out atmosphere, what is that? Way out there? The spacosphere? I just made that. I just lost a word, so I made something up. Anyway, there is a boundary outside of what is called what is known as the plasmasphere. And the plasmasphere is this cloud of energized gas that's outside of our planet, kind of outside of our atmosphere. And it's these gases that are electromagnetically charged, and so they've got these waves traveling through them, and they give off this hiss. It's like a static sound. So we can actually tune into it and hear the static from this plasmasphere. And it seems as though the plasmasphere, according to their research, could be interfering with these high-energy electrons zipping in once they've gone into the Van Allen belts and zipping in and hitting our planet. And they're like this plasmasphere is like a force field, and it just blocks them, so that there are a whole bunch of killer electrons that never reach our planet because we have this plasmasphere. That's pretty cool. But the researchers don't really know exactly what's going on. This is just their best guess. There's a hard, fast boundary, says one of the researchers. And so they have to get some new instrumentation and some new eyes to take a look at it and actually figure out what's going on. So there's a lot more to our magnetosphere and our plasmasphere and the outer atmosphere of our planet than we've come to think about. some neat stuff going on. Yeah. And then my third story that I just wanted to get in the beginning of the show is scientists took DNA, plasmid DNA. So plasmid DNA is DNA that's in circles. It's basically chromosomes that are circular in shape. And they took this plasmid DNA that had a couple of genes in it that they wanted to have specifically in it. They designed it specifically. And then they put the plasmid DNA on screws. And then they put those screws onto the outside of a ship that was launched up into suborbital space and then re-entered our atmosphere and landed back on the Earth. They took the screws out and they tested it to see if any of the DNA was there anymore. And they found out there was over 50% of the DNA that was left. And about 35% of that DNA was able to transfect bacteria and basically take the genes that they had added into the plasmid DNA and get them into bacteria and get bacteria doing what they wanted to do. So this study is evidence that DNA is very hardy and could potentially exist or survive through entry into our atmosphere and also the radiation of space.
So the DNA itself lasted, but we don't know what would happen with the process of DNA replication, right? Because that's kind of separate.
Well, we do know because they transfected some other bacteria so that they were actually, the DNA maintained its full biological function, 35%. So they were able to get back 50% of the DNA. 50% of the DNA in the screws survived. Mm-hmm. And 35% of that was actually able to maintain its biological function and to transfer, to copy itself and transfer into into a bacteria and get that bacteria doing what the genes were supposed to do.
Nice. Still not a passing grade.
Well, but it's just another little.
But it's enough.
Another little tick in the direction of panspermia's possibility.
That's exactly it, Justin. That's exactly it.
This is that whole idea that life could have started first on Mars, and a meteor strike there spread DNA somehow to planet Earth, which was just enough instructions to build the first mitochondria.
Yeah. If DNA can hang out in a screw and survive.
Yeah. But that's still, the power we've subjected all sorts of extremophiles to space, and they've survived. We know that they can survive in ice on this planet for hundreds of thousands of years when radiation should have destroyed them. Even in a frozen stasis state, there is DNA repair taking place. Now, I don't know if this study, of course, couldn't have been long enough to study that, but we have seen it in 100,000-year-old microbes that have their DNA self-repairing in that state. So this is opening up further and further the possibility that life, even without human intervention, could have traveled the star. Well, maybe not the stars, but the planetoids at the very least.
Right, and in this case, they went up on a rocket held in a bunch of screws. And if they could, and we know that if asteroids could have a nice little pocket for them to protect them in, maybe it would be that much better and easier. Who knows?
So Ed asked an interesting question related to that in the chat room. He asked, so no craft we send into space can be completely, truly sterile?
And we have talked about that on the show before. There have been studies trying to assess what is surviving in the clean rooms.
Yeah.
And things do survive on the walls of the clean rooms. It's I'm pretty sure that we're not killing everything.
So
I mean,
even if
this is-
we're not from panspermia,
We are
We might be-
panspermying.
yeah, we are panspermifying
Yeah.
Mars right now. And that
Pansperming?
comet.
we're putting our DNA into space one screw at a time.
Yeah.
Oh, yes, we are. This is This Week in Science. Hey, Justin, what you got?
Okay, so this is a statistic out of the National Turkey Federation, which is they say that 46 million turkeys are just on the verge of being devoured tomorrow.
It's a lot of
46
turkeys.
million turkeys. And you would think that maybe Earth is the most hostile place for turkeys, but they're not even safe in space. As tomorrow aboard the International Space Station, the in-flight menu includes smoked turkey, amongst the candied yams, green beans, mushrooms, cornbread, stuffing, and cherry blueberry cobbler.
Sounds delicious.
Yeah. So how could these things possibly be connected for a sciencey story? Let's see. Well,
Figure it out.
at NASA's request some time ago, University of Florida researchers were poised with the conundrum of how to turn human waste, the kind of thing that eating 46 million turkeys is likely to produce, into rocket fuel. So currently, I guess the waste on any sort of space station or anything is collected onto the re-entry vehicle in such ways that it is burned up upon re-entry. It's just waste; it just gets evaporated. But a while ago, back in 2006, NASA began making its plans to build bases, inhabited facilities, human colonization of the moon. And one of the things they didn't really want to do was build waste facilities to store it. They didn't want to just sort of have, open go. First thing we do on the moon is create an open sewer out of doors. No, they wanted to figure out a way to get rid of it, and because they were also at the same time working on the most lightweight vehicle that they could take back to Earth, to launch back, to return people to the Earth, the idea of collecting all the waste on the moon and rocketing it back and disposing of it in the way they normally had didn't make any sense. So they put out this problem. It was taken up by a professor at University of Florida, Pulamamon Apoli, who was then a graduate student. Oh, and then graduate student, sorry, Abesh Doli. And they were up to the challenge, and they've been working on this way to turn regular human waste into fuel. We are trying to find out how much methane can be produced from uneaten food packaging, and human waste, said Pulamana Apoli. The idea is to see whether we could make enough fuel to launch rockets and not carry all the fuel and its weight from the Earth from the Earth to the Earth for the return journey. Methane can be used to fuel the rockets. Enough methane can be produced to come back from the moon. NASA started supplying the scientists with a packaged form of chemically produced human waste. I think it's odd that NASA had to produce the waste. Like, there's no way. I guess just as a sort of problem to get a lot of waste to test. There's probably not a lot of legal ways that you can go about,
human waste.
Right, because I bet you there's if you run a porta potty sort of rental thing, I bet you have very serious restrictions. You probably can't sell that to the first researcher in a lab coat who says, I would like to buy 400 pounds of your finest end product, right? So NASA was providing the waste. How they got it, I don't know. It also included simulated food waste, towels, washcloths, clothing, packaging materials, a lot of the stuff they could see as being refuse on the moon. And they went to work. They found the process could produce 290 liters of methane per crew per day. And the result would be available in about a week. This result led to the creation of an anaerobic digester process, which kills pathogens from human waste and produces biogas, a mixture of methane and carbon dioxide, by breaking down organic matter in waste. In earthbound applications, that fuel could be used for heating, electricity generation, or transportation. So yeah, here's one of those things. We have a problem on the moon. We want you to try to come up with a solution, and we have now perhaps a plausible new process for energy creation here on the Earth.
Well, we have been using waste products to produce methane for fuel here on Earth. There are all sorts of waste treatment plants that use waste that pick up the excess methane, but not so many that actually try and process it to create methane more quickly. So I think that's
Quickly,
interesting.
efficiently.
Yeah,
They also
and there's the question of how much energy you're putting in to be able to get the methane out. Is that sustainable in the end? Are you ending up with a net gain of energy even with dealing with the waste? And so these are always things that have to be balanced out.
Yeah, this what they call this digestion process would also produce about 200 gallons of non-potable water per year from this waste. That is water held within the organic matter, then released through this decomposing process. Through electrolysis, the water can then be split into hydrogen and oxygen. Astronauts can breathe oxygen as a backup system. The exhaled carbon dioxide and hydrogen can be converted to methane and water in this process. So we're talking about separating out all the good stuff that goes down with the flush and turning it into things that we can use again on the moon.
I think it's a great idea. And if we can harness it here on Earth, if we can harness it on the moon, on the space station, wherever, to be able to actually deal with our waste in a way that we're able to gain energy from it, that it's efficient, that it's clean, that we don't have to use as much natural gas from the ground. I mean, that's great. That is great. And we should do that. So I really hope this is just as popular as Spam. Or more popular.
Now, hey, do you Thanksgiving's coming, lots of people may be going off to visit family. There may be a family member there that you've told yourself, even if the subject comes up, we're not having a conversation again on this subject. Because every time we do, it gets heated. Right? Mm-hmm. We also I know it's not very Dr. Spockian. It's probably not in any of the child rearing literature, but it turns out this is a recent study published in the journal Human Communication Research by researchers at Rollins College and the Pennsylvania State University. They found that individuals who are exposed to intense verbal aggression as children are able to handle intense conflicts later in life. Kind of makes sense, doesn't it? Right?
Maybe.
If you're used to a house full of everybody's yelling, there's arguments, and then you get into like an argument later on in life, you're like, oh, I know how these work. Yeah, you say this, you say that. It's just calm, just normal. I don't have to raise my voice at all.
But I wonder if it has to do with whether that verbal aggression was something they were engaged in themselves as children or experienced personally as children versus just watched happen. Mm-hmm. You know, whether you're watching your parents fight and just sitting in the, sitting back and watching it, or whether you are, your parents yell at you regularly.
Oh yeah, no, you don't have to yell at kids. No, I'm not telling anybody to go yell at your children. That's not what this is. Exposed to, meaning their parents fought a lot. They could see there was fighting going on in the household between the parents all the time, that sort of thing. They were better able to handle conflict resolution later in life, right?
Interesting.
And so what I'm saying is, if there's any children at Thanksgiving this year, and you're trying to avoid that conversation that might end loudly with a bunch of adults yelling at each other, go for it. Go for it. Win the argument. That's more. The kids will benefit from it. All the little kids at the table. This memory of this conflict will make them better at conflict resolution later in life.
Yeah, and it's probably like conflict is probably not yelling necessarily, but disagreements. So conflict simply means that there is disagreement and there is a serious exchange back and forth, and it's not just dealt with by one party turning around and leaving or something, but that it's battled out. That people work it out. So if you watch that happen, you have good modeling by mentors growing up.
Yeah, and probably if you think about it, if you're having these altercations, that means that people are dealing with emotions that they're having, and they're processing them and they're expressing them.
Mm-hmm.
And it's possible that if you have less of that, you have a lot more bottling up of emotions. Emotions are getting suppressed, and so those children don't know how to recognize emotions when they have them or recognize when other people have those emotions. So in terms of emotional intelligence, I think it makes a lot of sense if you're around people that speak their mind, that it helps you learn how your own emotions work.
Yeah, so you can deal with it better.
Yell in front of the kids. It's good for them.
Speak your mind.
Be emotionally honest.
I can sort of imagine. Can you imagine? I mean, I don't know what your childhoods were like in any way, shape, or form. There was a decent amount of yelling around the household when I was growing up. But I also sort of like wonder, like, because my kids really haven't seen a lot of that. And I kind of wonder, like, the first time they see people yelling, are they going to be like, "Bah! People are yelling. This is freaking me out. I gotta get," like, or are they gonna be like cool about it? Like, yeah, they're yelling. Like, they're mad.
It's an interesting question, but if they've learned personally how to deal with stuff because you and your partner are able to work stuff out without yelling, then they'll probably be cool with it. They'll be like, whatever. I don't know. Who knows? They'll be chill kids. I think it's time for.
Loves our creature, cry that small. 5 peg, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels that are both for her.
All right, let's jump right into some cladistics, shall we?
Oh, please.
I love cladistics. So cladistics are when you study how animals are related to each other in evolutionary trees, right?
And you make stick trees to draw those relationships.
It's so fun. So one mystery always in the reptile family tree has been turtles. My entire time in college and still up until this new study, turtles have been attached to the reptile tree with a couple of dotted lines. Scientists have never known where turtles belong. Either they broke off near birds and dinosaurs and crocodiles, the archosaurs, or they broke off where the kind of classic reptiles are, lizards and snakes, right? So they weren't sure where to put them. Did they go with them? Did they go with them? Did they go neither? Are they related? There was lots of confusion. A lot of this was because in the fossil record, turtles just kind of showed up. There was nothing like a turtle, and then all of a sudden there was a shelled turtle.
Ooh.
And
Maybe it's from like the aliens that the Scientologists have.
Turtles. I mean, turtles have been around long enough, right? That we, looking back through the fossil records, this is sort of diverged off of turtles. Like, turtles didn't even turn into anything else. They've just been turtles this whole time.
They've been turtles. So then when I was in college, it was really exciting. This turtle showed up in the fossil record that just had the plastron, the bottom part of the shell. And you could kind of see that the bottom part of the shell originated from the ribs. And it was this awesome thing where you're like, oh my gosh, here's some sort of clue as to where turtles came from. This is great.
But then they found the top.
Still, they did not find the top. Oh. Pretty cool. But still, no idea where the turtles fit in the family tree. Now, a new study out of the California Academy of Sciences here in San Francisco found out where turtles belong. They used ultra-conserved elements, is a technique of genetic sequencing that's new. This has never really been used before. Ultra-conserved elements, or UCE. And they used that to figure out more genetic data from turtles than has ever really been synthesized before. So they got this huge amount of genetic information, and from that, they were able to place turtles. And they placed them in a brand new group. And this is the largest group of organisms ever to be placed into a new phylogenetic group. So this is pretty cool. All turtles and tortoises are now in Archaeolosauria. And they are closest related to birds, crocodiles, and dinosaurs. Archosaur. So, yeah, which makes total sense because there are things that lizards and snakes share that turtles don't. Like there's like their watertight scales, for example. Turtles can actually breathe through parts of their skin. We've talked about that before, that they can actually breathe through skin near their cloaca. We talked about that before. And some turtles breathe through their mouth, and there's all sorts of weird things that they do with their skin. Their skin is semi-porous. That's not the case with lizards and snakes.
Aren't they also butt breathers?
Yes, yeah. So that's what I meant by the cloaca. Turtles don't really have a butt, per se.
They have a cloaca, similar to birds.
Yeah, exactly. But so there's that. And there's also their eggs. Their eggs are more permeable than lizards and snakes. And there's other things, if you look at them, that you can kind of see. You can see, this doesn't really look like it belongs in this group, really. It doesn't make a whole lot of sense. So they also have temperature-dependent sex determination, which means that dependent on what temperature the egg it grows to maturation in, it indicates whether it will be a male or a female. And that's usually found in crocodilians and turtles. There you go. There's another link between them. So this makes a lot of sense, and I'm super excited for it, because if I were to put my money on which group of animals they were closer to, it would have been these guys for sure. But now we have actual genetic evidence that shows us that this is mostly like where these guys will go. And now we have reclassified an entire huge group of animals, which is so awesome.
That's really awesome.
Yeah.
Next week, can you do me a favor and research, just for my own well-being, why hyenas are in the feline family?
Sure.
Thank you.
I'll give you an update on that next week. So that was my turtle. I think it's so cool, and it came out of the California Academy of Sciences. So
it's local, and it's awesome.
Yeah, and they did it by looking at current species, at frozen DNA from species. So they were able to kind of do this longitudinal study of all these different DNA samples with this new method and find out this new information. So there we go. Moving right along to frogs. Would you guys guess that frogs like to hang out in one place, like that they like to move around? What do you think?
I think they hop around.
You think they hop around? So they are generally considered to be sedentary. They are generally considered frogs to hang out in kind of one spot. They're not migratory. They don't move in large areas, right? So
unlike the salamanders and newts, there's a giant newt migration happening in the East Bay currently.
Oh my goodness, watch. Break for newts.
That's right.
But so scientists have always thought that frogs don't have a great sense of direction, that they just kind of know, here it's wet, here I'll stay. Well, you guess what's coming? Frogs are smarter than we thought.
Woohoo! Duh.
Yeah, right? Yet again, an animal is smarter than we thought they were. So looking at brilliant-thighed poison frogs in the University of Vienna, they put these little frogs, they put teeny tiny transmitters on them that allowed the scientists to track the movements of these frogs. Now, these frogs need to move around because they live in leaf litter. They don't live directly in the water. So they have to move from pond to pond. And their tadpoles, they actually have to move from pond to pond on their back. And the tadpoles can't get too dry, and they also can't move around on land because they don't have legs yet. So they have to know exactly where they're going to get to these ponds. So by putting these transmitters on them, they then put the frogs in different parts of this forest, and they watched how well they did in finding their way back to the ponds where they lived. And what the research found is that if the frogs were placed within a certain radius that was familiar to them, they went straight back to their home. They didn't pause, they didn't look, they just marched straight back to that pond. But frogs that were placed in an unfamiliar terrain got lost, wandered around, had a real hard time getting back. Eventually got back, but it was very difficult.
So, now I want to know what'll happen if we put them on top of a meteorite.
So they have landscape.
You just made my day.
They can recognize this tree from that tree. If they've been there before, they're like, okay, I know where this is. If I follow that log past that tree, my swamp will be on the left.
See, Justin, we don't know that, though. They could be using physical markers, or they could be using chemical markers. There's any number of things that they could be doing, and we don't know what it is yet. All we know now is that these frogs have what the scientists have tested as up to 100-meter radius from where they were.
Which is all the researchers have done. They just took them a maximum of 100 meters away. They probably haven't gone any further.
Yeah, and prior research has shown that they have been able to go as far as 600 meters.
Yeah.
So in this research, they found 100 meters for sure these frogs can handle. And so that's the question, though. I mean, we've been asking for how long about how pigeons get from place to place. Now we have to ask, how do these frogs know where they are? Is it chemicals? Is it markers? Is it magnetic somethings in the brain? Is it gravitational
gravity? Yeah.
Could be any number of things. Yeah, so it's pretty wild. And then the last one that I have, pretty quick one. Mining. Good or bad for fish? What do we think? Good? Bad?
Mining, bad.
Bad.
For fish?
Thumbs down.
For
Yeah.
fish, yeah, can't be good for the fish.
The stuff that we use in mining, the mine tailings, all the stuff that comes out of separating the stuff we want from the dirt,
Yep.
bad.
Yeah, so there's
for 100
all sorts
years
of
we
stuff.
just used mercury, mixed mercury in with water and dirt to separate the gold. So
Right.
There's all sorts of things going on. There's the chemicals that you're talking about. There's also the fact that they use water that they usually pump
lots of water.
out of the rivers
Oh, yeah.
Hydraulic mining.
loosen up the sediment
That's what they mined with, was the water.
that then runs back into the water that's murky, right? So there's all sorts of things going on that influence the waterways directly next to a mine. New research has looked at how widespread the effects are. And a mine can affect areas miles away, sometimes even in a different state, sometimes something that's not even directly connected to the waterway that the mine is next to. So there is a huge devastating impact from these mines on fisheries. And that can be from mining things like, one of the most common ones is coal, of course, precious metals, pebbles, sand, salt. All these things mess up the environments. They're messing up the flow of streams. They're reducing the availability of trees to grow. And they're compacting soil. And all of that changes fish habitats. First of all, the fish have to be able to see when they're in the water. Second of all, they have to be able to lay their eggs, because a lot of these are in streams where these fish are laying eggs. And if the sediment's not quite right, the eggs will get washed away or they'll get buried. And so there's all sorts of problems with mining. And so usually an environmental impact statement is done for the area right next to the mine. But this new research has shown that the spread of the effect of that mine is much larger, which hopefully will mean that perhaps someday the environmental impact report will have to reflect a much larger distance of impact from that mine.
Yeah, no, that would be good. I think that there are a lot of things that we need to start considering in this kind of cradle to grave perspective. You know, what is the pathway? What is the ultimate What is where does it start? Where does it end? How big is the area of what is affected?
Hmm. I also like it because it's just another reason that coal's not that great.
Yeah.
It's an interesting thing though that was sort of coined in there. It could have effects on unrelated waterways. And at first I was like, well, how is that? And then it dawned on me, yeah, if you knock down the fish population in one waterway, and you still have the same amount of fishermen, they're going to go where it's a little more plentiful and have a bigger impact fishing an area they might not normally, that sort of thing. So
Definitely.
sort of
Plus,
cascading effects.
Or
yeah.
if there are things like in this image that I'm going to share from the news release where acid is seeping out of a coal mine into the waterways, that acid is going to go from one small waterway to another waterway. And although larger amounts of water will have a diluting effect, there is still an increasing amount of acid entering the waterways. And like you said, the populations of fish are going to be affected all the way down.
Hmm. And it's really hard to predict how water is going to move. So it's possible that this acidy water will flow into a larger tributary, but then that tributary might separate again. And it's possible, depending on the way the water moves, that one of those forks will get all of the acid.
Mm,
It's hard. It's hard to predict, especially if there's different temperatures of water mixing or
right.
different
Yeah,
types of minerals that are mixing in the water. It's not always going to be 100% dilution. It's not going to happen that way. So that's really hard to guess. It's also true that if you destroy spawning grounds in one area, those fish, if they hopefully figure out to go somewhere else, they'll move into another area that may crowd other fish, or it might reduce genetic variation among the fish if all of them have to go into one area.
Yeah, and lead to a bottleneck.
Yeah, there's all sorts of weird stuff going on. But I think it's important to remember too that all this stuff that we have, like coal that we burn for electricity, there's all of the obvious stuff that's bad about coal, but there's this other stuff too, that where does the coal, you have to kind of look, it's what you were talking about, the whole story of this piece of coal, from when it gets pulled out of the ground to when it gets burned to when it gets turned into electricity. It's this long story, and there's all sorts of little stop points along the way that have different impacts.
Absolutely. Mm-hmm. And on that note, it's time to take a break from these brain impacts that we're giving you with this information. This is This Week in Science. We're going to take a break. We'll be back with a lot of stories about your brain, how your brain works, what your brain is like on imagination, and much more. So stay tuned for more This Week in Science coming up next. This Week in Science is made possible by listeners like you, and this hashtag Thanksgiving, this Thanksgiving, I want to say thanks to all of you who are making what we do possible, who are making our TWIS shorts that much more possible to get done and get edited, to make our weekly shows that much better by giving us the availability to be able to make changes and make things happen. It's all because of you, and I hope that what we do as we move forward is making the show better for you. I really do. Thank you. No matter what, thank you so much. And if you do want to support us this holiday season, there are a few ways that you can do it. The first, you can buy our merchandise. It's gift-giving season, right? We're almost coming up on Twistmas and Twisma Kwanzaa Hanukkah and all of those. So why don't you look at the TWIS swag that's available in the Zazzle store. Go to twis.org and click on the Zazzle store link. You'll be able to see all the stuff that we have available to your favorite TWIS minion. Check it out, twis.org, our Zazzle store link. Seriously, go do that. Buy a gift. It'll be good. You'll love it. If that's not what you want to do, however, we also do accept support in the way of donations through PayPal and also Patreon. We have PayPal links all over our website, twist.org. So if you head over there, you'll see PayPal buttons on just about every page. And Patreon, you can go to patreon.com/thisweekinscience and choose the level of your support for per episode. You get charged at the end of every month, and it goes on an ongoing basis for as long as you would like to continue supporting us. Makes it really easy. It also is a fun way to become a part of the show. I do hope that you consider giving to us this season through PayPal or Patreon. Patreon.com/thisweekinscience. And if money or stuff isn't your thing, well, we would appreciate you telling your friends and family about TWIS. We could always use some more listeners, some more YouTube viewers. Whichever way you enjoy TWIS, maybe your friends and family would enjoy it that way too. Put a shout out for TWIS up on your favorite social media network the next time you listen to us or watch us online. We really would appreciate it because you know what? We couldn't do this without you. Thank you for your support.
Explain the things you heard with more than intuition. The line of reason shows the way to go. No conclusion. The methods of hypothesis and patience are the only.
And we are back with more This Week in Science.
Oh yeah, Justin, tell me a story.
What have I got? What's next up on here? Oh, this is the ambulance story. This one cracked me up when I read it, and not for all the right reasons. But This is a study where they looked at ALS versus BLS. ALS is the advanced life support, which is most ambulances these days actually are, at least probably in the Bay Area, probably in this area too, the advanced life support. Meaning they've got sophisticated gear, staff of the crew, highly trained paramedics who are ready to administer all sorts of treatments right there on the spot, including intubations, IV interventions, that sort of thing. This is the cutting edge modern era of ambulance that's out there. Interestingly, though, they did a study of 380,000 cases of people who had out of the hospital cardiac arrest, and it said 90% of which, when they get to the hospital, don't survive. And they found that of those that do survive, they're 50% more likely to survive if they had the old school basic life support ambulance versus the advanced life support ambulance ride to the hospital. And sort of the drill down on this, if you can imagine, seems to be that with the basic life support ambulance, they're not equipped. We can't handle this. We need to get them to the hospital as quickly as possible so they can start administering treatment. Whereas in the advanced life support, the theory is perhaps they're trying to do too much before getting to the hospital and not focusing on just getting to the hospital as quickly as possible. So if you do get into one of these arguments during Thanksgiving, and that relative you were trying to avoid the conversation with, but it couldn't help it, crept in, and you had it anyway, and you had the time, there was yelling at the top of the lungs, and then they went and keeled over. When you call the hospital, ask for the cheapest, most basic ambulance that they've got. The family's going to look at you funny. The family's going to think you're trying one more thing to get at this relative who obviously you're not getting along with. But in fact, you're increasing their odds of survival.
That's interesting. When it comes to the line of how long it takes before you get to the hospital, and if that is in fact the the line, if that is the thing that really does determine survival or not. Just get to the hospital as fast as possible. But it's an interesting result. It's a very interesting result. These things this came out of the journal JAMA, Journal of the American Medical Association, Internal Medicine. Get thee to the hospital.
Get to the hospital quick. Also,
as Dave Friedel says, the people who are working the ambulances are paramedics, and they are not doctors. So there is a potential difference there in the treatment, the quality of treatment that you receive during that time that you are receiving treatment.
Right.
And also, disclaimer, we are
maybe
not
a hand-pumped ventilator, where the other one's taking more time to intubate you properly and get you on some sort of more. It just takes more time. Everything takes more time. And what basically the essence of it is, time is of the essence. Time is the important factor between getting to the hospital, not anything that you can do to try to stabilize on getting there. This is wild, though. As we're about to eat 45 million birds, put 45 million down, a new bird species has been confirmed. This is a new bird species that they first witnessed, I guess, 15 years ago, and it's taken that long to find another one and really identify it. It was in the Indonesian island of Sulawesi, newly named Sulawesi streaked flycatcher. Distinguished by its mottled throat and short wings, was found in the forested lowlands of Sulawesi, same place basically where it had been first observed. Researchers report this in PLOS One, Public Library of Science Online, that the new species is markedly different from other flycatchers in its plumage, body structure, song, and genetics, proving that it is a new species unto itself. Because the bird has survived in a region heavily degraded by cocoa plantations, the species is not currently at risk of extinction, but may be in the near future.
I think it's interesting that this study is also using genetics to confirm the new species compared to others. I love that that's becoming more of a staple of species determination. I love that. I think it's time to start using that more so than physical characteristics, because we know.
Oh, absolutely, and it is becoming. I mean, there was this theory that a BBC documentary put that the first Americans in South America were Aborigines from Australia, based on some morphology and somebody who was claiming to be a primitive cave art expert. These sorts of theories are all possible. You can make-itty up them, fill in dots, use some morphology of a skull you find, but DNA, genetics, that's where the truth is gonna be revealed.
Species are never going to be a perfect thing. There's still lines we're drawing in the sand that in the real world don't mean a whole lot, but definitely DNA is going to be the best way for us to be able to standardize it at all.
Yeah. And another kind of sideline to this bird study that you found, there was another story that I found in which researchers were looking at diversification and speciation in birds, and they found that a stable habitat is actually a main player in speciation. So if a habitat is changing too rapidly, it actually diminishes time to speciation. The longer a habitat is, or an ecosystem is stable, the more species that will come out of it. And they looked at different birds, like ground-dwelling birds and birds that were flying. So ground-dwelling birds and treetop-dwelling flying birds, they found that there are a lot of species of these ground-dwelling birds. They were speciating among themselves, and there was also a lot of speciation among the tree-dwelling birds. However, when the trees were disturbed, then it decreased the amount of speciation. So our humans, as you mentioned, the coconut, was it cocoa or coconut plantations?
Cocoa. Yeah.
Cocoa plantations for this bird, it's been able to hang out in these plantations for a while, but the more that we mess with ecosystems, the more that we decrease populations' ability to adapt and speciate and possibly deal with change.
Which is wild because it's almost the exact opposite of what I would predict would happen.
Yeah. Yeah, it is. You'd think that this is one study, so there could be different situations where it works the other way, but
isn't that what you expect? Evolutionary pressure is going to be driving these changes.
But it's not so much. It's the
environmental pressures changing or creating evolutionary pressure.
There is that
change.
Right, there is that, but what they're looking at is the competition between individuals in populations is going to force changes in certain directions to be able to speciate to or adapt to be able to take advantage of a different niche in the ecosystem, to be able to take advantage of another food source, to be able to take advantage of something that doesn't have as much competition for it.
Yeah, which retrospectively, this is really interesting, because then retrospectively, through the record of evolution, that thing that we can say, because the trees were gone, they were forced this life form was forced to forage more on the ground, may be that the one who, when nothing was happening, was using the niche of the ground, which wasn't as popular to forage on, and when the environmental change took out the trees, they're like, they flourished because there wasn't anything else to compete against, and they're the one thing that we know went on and moved forward. That's very curious.
Yeah, it's all very curious. Yeah, but it's out there. I should need to actually find it so I can post the link up. But it was now
it's gone from not making sense to making perfect sense. That's the way it's always been. Right? Of
course. Well, that's the way our brains work, right? That's the way we integrate things. So researchers have used high resolution, functional magnetic resolution, to be able to pinpoint memory being formed in our brains. They took a bunch of people, like 20 or so individuals, college students, and stuck them in these 7 Tesla, like really big magnet fMRI machines doing 1 millimeter resolution, so tiny resolution for what they're doing. Normally the voxels, or the size of the pixels that they study in fMRI, are not as small. So this high resolution imaging is like, it can really pinpoint small areas of the brain. And so that's what they did. They had these individuals look at images while they were in the machine. So there were new images that they were seeing while their brains were being imaged. And then they kind of tested them later. So had them look at images that they'd seen before. And they found that in the brain, the hippocampus of the brain, which is like kind of deep in the middle of our brain, the hippocampus is that information comes into it through an area called the entorhinal cortex. So they saw activity in the entorhinal cortex and then in the hippocampus when novelty was being perceived. And then when something was familiar, they saw stimulation or activity in the hippocampus, in the CA region of the hippocampus, and then flowing back to the entorhinal cortex. So there's this information into the hippocampus pathway, and then there's an information out part of the pathway as well, which is pretty interesting. And this is something that has been theorized but has never actually been shown before. So to actually be able to get the resolution to be able to see this activity, this activation within the brain is pretty amazing. So we now know that information does actually go into the hippocampus. The hippocampus is definitely active and pretty much responsible for, or this doesn't actually it doesn't show that it's responsible for it, but it's active during memory formation, during a novel stimulus being experienced. And then
Is it something like a distribution thing where it's like information comes in, it's like, oh, we don't know what this is connected to. We don't know where this goes. Let's keep it here. Figure out a pathway out where we can have a destination for it. Where something you see this every time, this is a familiar face. That information comes in. Oh, we know this. It's related to these. Let's send it out and let those either store or connect to it.
And so memories are known to be stored or thought to be stored in the cerebral cortex. They're also stored in the cerebellum, but a lot of stuff happens in the cerebral cortex. And so it's thought that there are these different layers of the hippocampus, and there are what are called, in the hippocampus, there's an area called the dentate gyrus, and the dentate gyrus is connected to these areas that are called CA1, CA2, and CA3. And it turns out that CA1 seems to be kind of like a novelty detector, and so it's kind of like the layer of the hippocampus that's going, have I seen this before? Is this something I've seen before? And it's probably talking to other areas of the brain to determine if this is something new. And if it is something new, then information is brought in entorhinal cortex to the hippocampus. If it's not something new, it goes from CA1 out to entorhinal cortex and goes back out.
So CA1 is either referencing the whole rest of the brain.
Yeah, it's like it's a reference
layer. Recognized.
Mm-hmm.
Or would be even weirder, it has a microfiche blueprint of the brain and things that are already there. Right? Because it's not, how else does this filter work? How else does this filter know?
Yeah.
Whether it's a hit or a miss.
These are the bigger questions, right?
Figure out what to do with it, or it's like got a list of everything that's come through.
Yeah.
That's crazy.
Yeah. And so things get into our brain, right? And so you've got these viewing of things, and your hippocampus becomes active, and if it's something new, then it decides to hold on to it and store it and shunt it elsewhere in the brain. And so another group of researchers at the University of Wisconsin-Madison used this very high-density EEG to be able to determine which direction information was flowing in the brain during either watching a movie or during the experience of imagining or trying to imagine what they had seen previously in a movie. And this high-density EEG is pretty insane. It's like all these little sensors, and they have a cap that basically covers the entire head, so they can figure out where they can triangulate, but using all sorts of points of the triangle to be able to figure out where the signals are coming from. They determined that in our brains, we are getting information from the, let's see, where did they say? They said it goes from when we're viewing something, it goes from our occipital lobe, so kind of like our visual cortex, our occipital lobe, to our parietal lobe. And the parietal lobe is a lower order region, or a higher order region. And so what ends up happening, it goes from lower vision in up to a higher order region where the parietal lobe, it starts combining all that stuff with other things that we know, other senses. And then when we imagine things, they found that the flow of information goes from the higher order, from the parietal lobe, down to the lower order, the occipital lobe. So it goes from the area where it combines the senses to the lower order region, which is where we can just imagine the visual scene. So there is a difference. One of the big things about this is that now we know there is an actual difference between seeing something and imagining it. Our brains do these things differently. There are different pathways, or it's the same pathway but backwards. So even though we talk about imagining doing things in order to teach ourselves how to do something, or that our brain, it's as if, our brain is, I've read things before and kind of parroted this information myself, that our brains, we live in our brains, and so if you're imagining something, it's as good as the real world. It's not. Our brains know the difference because the information flow is completely different.
There you go.
Yeah.
So it's not all like the Matrix.
No, it's not all like the Matrix. And speaking of the Matrix, you just gave me a perfect segue into my last brain story. Speaking of the Matrix, the brain knows the difference between the real world and virtual reality. So, research looking at mice, they put, I think it was mice that they did, they put them in a virtual environment where they basically they put them on a treadmill. Oh, they're the rats. This is rats, not mice. Here we go. They put them on a treadmill so the rats could walk forward on the treadmill. And then they surrounded them by screens on which a 3D environment of a room was projected. And then all the lights were turned off in the room so that really they were just surrounded by this crazy, what we would think of maybe as like an IMAX experience, but way more intense, like as if they were in a room, right? More real than an IMAX experience. And they stuck them in there, and then they looked at the brains, looked at brain activity while they were walking around this virtual room. And then they compared it to what was happening in their brains while they were walking around a real room that looked exactly the same. And they found that there are the hippocampal neurons fire totally differently. So in the virtual world, hippocampal neurons almost fire randomly. Like the rats don't really have an idea where they are. They behave normally, and there are cells in the brain that are called place cells that kind of tick off space and different places in space. And these cells instead, and they integrate all sorts of sensory information from our visual, auditory, vestibular system, all sorts of things. And by taking away a whole bunch of stuff with the virtual world, all that the place cells seemed to do was tick off distance. So they just ticked off how far the rats walked on the treadmill. Whereas in the real room, they were like ticking off all sorts of information about places in the room.
Interesting.
Yeah. So these are rats and mice. The only human study was the hippocampus memory study. That was a human study, so that's what we do know. But what this implies is that for all this virtual reality stuff where people are really worried about, how it's going to affect our brains and, how people are going to get confused about being in the virtual world versus being in a real world, our brains know the difference.
For now.
Pessimist.
I am optimistic about the level of technology that we will have in the future.
Great, yeah.
That if it gets so good that it becomes tactile, or it finds some way to incorporate elements of the real world besides just visuals, that's where our brain could start to get confused. I feel like if you incorporate multiple senses, well, besides the two, I guess, we do vision and we do audio cues, right? We have those often in virtual reality. But if you start to add the rest of them, it is possible that you could start to confuse your brain, right? I mean, that's what it sounds like to me, is that we just haven't gotten convincing enough yet with our virtual reality to confuse the brain.
Fair enough. Fair enough. I'll agree with that.
So for now, we're safe.
For now, we are safe from the virtual worlds. Our brains know the difference, and it's really interesting that there are actually that the cells in our brains, certain cells in our brains do, or in rat brains, and we would guess that in human brains as well, but we don't know. But they fire differently. They don't. are not as activated by virtual reality at this point in time.
And very likely the only way to make a very convincing virtual reality is to start an individual, be they rat or man,
in virtual reality and have them live in it.
and raise them within it.
Raise them in it. Mm-hmm.
A Truman Show situation.
Yeah.
What I'm wondering is, if you combine those two stories that you just talked about, that's what's really missing in the human element is the imagination, right? So the difference between the rat and virtual reality.
Are you saying rats don't have dreams?
I'm saying that the rats don't have a personal motivation to trick themselves. But humans sometimes do. Humans get caught up in the fun of imagination and pretending to be somewhere they're not. And it's possible that we would see different things going on in a human brain in virtual reality just because of that. Because we are able to suspend disbelief and get caught up in things because we have such a powerful ability of imagination. Now, from what I just heard, though, imagination is a vastly different area of the brain from real world experience. So if you add those two, in theory, it should still be totally different.
In theory, yeah.
Theory.
Imagination uses the same areas of the brain, but in the opposite order. So it should be different. In theory.
Mm-hmm.
Yeah. So
you need to use that giant chainmail helmet situation.
But hang on now.
And
If we're
put
talking
someone
about
in virtual reality and see what happens.
If we're talking about virtuality, imagination is just filling in the gaps. Imagination is just covering up the mistakes at this point. Imagination is just making up for the deficiencies because the virtual reality itself would be following the same pathway through the brain as reality. It would start with the lower order, the visual intake, and then move up, and not be a projection back down the other direction, unless you're trying to cover up, well, you know, the dinosaur didn't look that real, but I'm going to go ahead and cover up and suspend the disbelief to help it be a little bit more real, right? Which is sort of what you were saying.
Right, which is something that humans might do.
Might be able to do better, yeah.
Yeah, might be able to do. Any more stories out there, everyone? Justin?
I'm good. I've got an orange I'm about to eat, so.
Okay, Blair.
I have something really cool. If, Kiki, you are able to pull it up on the laptop so we can hear it, because it's audio.
Oh, yeah.
So
I will do what I can.
All right. So everybody's favorite, the peacock jumping spider. So this is not the peacock jumping spider. It's very closely related. It's another kind of jumping spider who also dances for its prey. Not for its prey, for its female. The males dance for the female. But they also do a complicated audio cue for the female to let them know that they're eligible and sexy. And it is wild. They do it by vibrations through precise, varied leg movements. So they are basically playing the drums with their various appendages, and it sounds awesome.
I'm working on finding it.
So it's Habronatus ciscatus is the genus and species of this jumping spider. Oops. So, and it's considered a multimodal signal to the females during the courtship display because of the dancing with the sounds.
Here we go. Let's see if we can get this to work.
That's just me. That's just me on the table. That's not the...
This is an old laptop. It's taking a long time to load.
Oh, no.
Okay, there it is. I can skip an ad now, maybe.
Maybe.
Whoop. Nope. Science!
It is science.
It is science. Here's a new spider.
They're
Doing a
in the
different
jungle.
jump.
That's got to be at least 180 beats a minute.
So these vibrations are measured with a laser vibrometer. They actually use a laser to measure these vibrations, and then they turn the vibrations that are measured into a sound that you can listen to.
Right.
This is not something that we would ever be able to hear otherwise.
But the female spiders can hear it.
The female spiders can hear it.
And it's somewhat comparable to this. You can kind of get the idea from it, which is great.
Yeah, and so right now what the spider is doing, he's got his back legs way up over his head, and his front legs are going like that. They're vibrating around really fast. This is just a spider drumming.
Bring us to this GIF. Let me see if I can do it.
Wow. That was a pre-mount motif.
Yeah.
Le Grand Spider Finale.
Le Grand Spiders. Yeah, these researchers are out of UC Berkeley who do this, and yeah, they do some cool stuff. I actually spoke with a grad student who does work on this, and we should get her on the show to talk about it.
Let's do that.
Yes.
Oh my gosh. Look at that little spider playing the bongos.
Blair has an image of a little spider, a little meme. I shall play you the song of my people. That's pretty funny. Somebody gave a spider bongos. I don't know if that's a good idea.
So anywho, that was my amazing, another reason I love jumping spiders.
I love it because
They're
they are complex and they drum songs to their females. Quick news. Researchers have been cataloging the microbial genes in our gut. And right now the catalog features nearly 10 million microbial genes. It's a lot of genes. 10 million microbial genes. And so these will give us information on how they influence human health. And most of the genes, around 6 million, are shared by only 1% of the population, which is, I think, the most fascinating part of that.
Oh.
Yeah. And another interesting study, researchers are proposing that the Higgs boson that was found, researchers got the Nobel Prize for it and everything, that it might not really be the God particle that everyone was looking for.
Say what?
Yeah, well, they say that, yeah, sure, there's a particle there at that signal, the 125,
Gigawatts.
Yes, the signal that they measured, that there is something there, but it's not necessarily the Higgs boson. And what they are discussing is a competing hypothesis to the standard model for physics, which is called the Technicolor model. And according to the Technicolor model, all of the little particles that make things up are probably made up of other little tiny particles. And so the Higgs boson is probably a particle made up of other little tiny particles that are held together by an as yet undetermined force. And we need a bigger atom smasher to be able to determine that.
If I just stuck to my guns 6 more months, my protest might have seemed relevant.
That's right. Yeah, but if you're interested in looking into it, the Technicolor hypothesis is actually the model that has been proposed is actually very interesting. And while evidence is still supporting the standard model, that doesn't necessarily mean that it doesn't support this Technicolor idea as well. We just possibly haven't had enough energy, high enough energies to be able to see to that point as of yet. So this is something that is still wide open. Anyway, I think we are done for the week, everyone. Yeah? We're about done here?
Yup.
Yeah.
Cut it out.
we are going to shout it out. I would like to take this moment to say thank you to everyone who listens to and watches This Week in Science. If you are watching right now, I am currently trying to log into Patreon so that I can actually read off all of our Patreon sponsors. But everyone who listens to and watches, whether or not you support us on Patreon or in other ways, thank you for listening. Thank you for watching. This is Thanksgiving, and the fact that you are giving us your time and attention means a lot. Thank you so much for that. And for all of you who are supporting us on Patreon, thank you so much for your financial support. It does help us do all sorts of things. And I'd especially like to thank Kevin Donald Wesley Ballard, Paul D. Disney, Sam Winsier, John Ratnaswamy, Rudy Garcia, Steve DeBell, Stephen B., Jason Dozier, Matthew Litwin, Eric Knapp, Andreas Akestad, Jason Roberts, Patrick Cohn, Shane and Tara Ginsberg, Byron Lee, E.O., Bob Calder, Jared Glysett, Ulysses Adkins, Jake Jones, Nicholas Lai, Brian Hedrick, Cassie Lester, Chris Cross, Sarah Chavez, Stephen Srowiak, Layla, Paul Stanton, Marshall Clark, Charlene Davidson, Henry, Don Komarechka, Randy Mazuka, Ed Dyer, Tony Steele, Lord Trentonian Moody, Larry Garcia, Daryl Lambert, Brian Condren, Richard Bowersox, Kevin Parachan, Dave Measure, Brian Rossman, James Dobson, Steve Goodwin, Kurt Stefan, Michael George, Craig Landon, Russell Jensen, Mountain Sloth, Jim Drapeau, Darren Lute, Tara Payne, Alex Wilson, John H. Maloney, Jason Olds, James, Paul West, Lauren Lang, Ed Bennett, Thomas Mikanen, Alec Doty, Illuma Lama, Nick Gradwell, Dougal Campbell, TMRO, Miko Pakala, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie Scott Lajewski, Tyler Harrison, Ben Rothig, Columbo Ahmed, and Gary Swinsberg. And thank you to everyone who is supporting us, even just $1 an episode or whatever level of support, and hasn't asked for any thanks at all. Thank you. I don't care if you don't want to be thanked. I'm thanking you right now. Seriously. Thank you all for your support on Patreon. If you are interested in supporting us, you can find information at patreon.com/thisweekinscience. Also, remember that you can help us out simply by telling your friends about TWIS. On next week's show, once again, we'll be broadcasting live online at 8 p.m. Pacific time, Pacific Daylight Time these days, on twis.org/live, where you can watch and join in our chat room. There's fun stuff happening in there. There's a lot of fun things going on in there all the time. And don't worry if you can't make it live. It's okay. You can find our past episodes always at youtube.com/thisweekinscience or at twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile device, you can look for TWIS for Droid in the Android marketplace, TWIS in the Apple marketplace, or This Week in Science in the iPhone podcast app.
For more information on anything you've heard here today, show notes will be available on our website. That's www.twis.org. That's TWIS.org, where you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for some more great science news.
And if you have learned anything from today's show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science.
this week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
This just in. This just in.
It's
Hi.
the after show. Hi, everybody. I wanted to place. Oh, I don't know if I'll be able to play something. Maybe I'll just share it. Maybe I'll just share it in the chat room.
Here, I'll share this in the chat room.
Something special in the chat room. Why did this not go up? What are you sharing in the chat room?
Oh, nothing.
No, just a little nothing.
Good science to you. This is Blair Basdrich for This Week in Science, coming to you from Aquarium of the Bay on Pier 39 in San Francisco. Let's go inside, see some fish, and talk about some science news.
What's happening? What's happening this week in science?
The olfactory portion of it.
Haha, it's a teaser. It's up. It's up on This Week in Science. Our most recent TWIS short is published, everyone.
It is.
It is.
I got a chance to see it. Awesome, awesome backdrop, by the way.
It's
For the
beautiful.
shot. That was brilliant.
Yeah.
Where is it?
High production value.
It is
I
high
think
production value, right? It looks great.
Where is it?
I'm very
On the
proud of
phone.
my editing, too.
Where can I find it?
I just put the link in the chat room.
Oh,
That's not the link I put in the chat room. That's something else.
What did you put in the chat room? It does. Thanks, Ben. The new short rocks, Blair Baz. Woohoo! She's going to watch it now.
I am going to watch it now.
4 minutes of aquatic science.
I'll multitask.
Heaven. In a world where you have 4 minutes to watch.
I'll have to send this to our shark guy.
Yeah. Yes, it is up. It is ready to be viewed. I put it out late last night, so I could definitely do some more promoting of it. I haven't done that yet, but
Wait, I don't see the link in the chat room.
I just posted it. If you
I
look,
see it.
it's right underneath yours, whatever yours was. What was yours?
Mine's what happens when you eat turkey. You eat a bunch of food for Thanksgiving.
You get a round belly.
Yes. All right, I'm opening.
Making...
I watched this earlier, but I'm gonna.
I want to watch an ad.
How come I'm not subscribed to this? Oh, now I'm getting redirected. I don't want to get all redirected. I just want to subscribe. Leave me alone. Don't make me watch stuff I'm not trying to watch. Oh, there it is.
Why do you feel so full after a big meal? Because you're full. It's gas also. I'll watch that video later. You always have to have a towel. Are you going to take your towel with you, everyone? I always have a towel. You have to always have a towel. Who's watching it now?
Me.
You're watching it again? I think Justin's
Again?
watching it. Justin's watching it too. I was like, I hear it somewhere. It must be in his monitors. I hear
Can
it.
you hear it through? Oh, you
Yeah.
can. Oh, sorry. Hang
I
on.
hear it.
So,
Am I feeling inspired with all my daily inspirations? Yeah, actually. It's been good. I mean, I don't know. It's been a total challenge to do something different every single day. And some days I'm like, oh my gosh, I don't think I can do this. And some days I haven't been able to do things, but I've tried really hard most of the time and to fit something in somewhere. And so it's nice. I really like it because there's a certain part of me that thrives on organization and having scheduling and making life a certain way. And then there's a certain part of me that's like fights against it and is like, I need to get out and do other things. And that's the part of me that needs the other stuff for inspiration. So it's kind of cool. It's working for me anyway. And it's like something every day that I have something little to look forward to every day, something new and different every single day. So my daily inspirations are working for me very well. If other people are doing them, I hope they're working for them too.
my daily inspiration right now is trying to train my 11-year-old to edit audio.
Nice. That's cool.
Yeah, I've given
Little
him
audio editor.
a bounty for him. If he can pick it up and do some editing of the radio plays.
Right. Oh, I still have to
Enhancing.
record for you.
Yeah, what happened, Justin? Do we have to re-record that whole episode?
Yeah, that whole thing is unusable.
Okay. I'll come up again. That's
Okay.
fine.
All right. Yeah.
Davis is fun.
Yeah.
I may not even go studio next time. I may just try to do all
Studio?
the recording here at the Moonbase studio.
Well, I have a steering wheel that I can bring of my own.
Oh
So-
yeah. See, I don't know that will help. I haven't
Really?
figured out. I haven't figured out a way to do 2 USB ins to 1 USB out for audio yet. I haven't found that mixer. I don't know if that exists. I don't know if that's a real thing.
Oh, yeah. 2 USB
Right.
mics
Into a
in.
mixer would be the idea, into the recording of the audio.
Mm,
And so I don't know if that exists. I still have some old school mics
no.
around here that I could pop into a mixer that I've already got. That would
I
do
have
the
a normal mic with a normal mic cord.
Yeah, so now that may be the way to go. And I've got
Well, I
more
have
than
that. I have a really
Yeah.
nice microphone from my pub quiz days.
Okay. And I've got my original TWIS mic still, and that might right there be the recipe to make this work. And I've got a little 2-channel mixer here that would be enough.
You could do it.
And again, I mean, so far the only thing I've got. No, it's not even. Gosh, it's not even that. See, I thought that we were gonna get that knocked out. Oh wait, no, it is a cat. Tony Steele. Tony Steele's got the only decent recording for that episode yet, for his part. His part's beautiful. It's just, he actually is playing 2 different characters, differently enough and amazingly enough that those two parts are locked down. He's my new go-to guy for the one that if I ever can get to this again to follow. He's definitely getting several roles.
Nice. I imagine he would have a great voice.
He does have. He has a great voice and a good command of being in character.
No, I know he has a good voice. He's been on with us before. Yeah. Being in character. Yes. I'll record mine. I have to put time aside and record mine. I can get you one good copy of my
The
script.
last one was awesome. I actually ended up using, instead of sort of editing you into stuff, because your copy was pretty clean read-through pretty much all the way through, whereas all the other versions were done in sort of fits and starts, because I'm getting 3 people in the room, here's what we're doing, blah, 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 and then that part's all getting done. Yours is linear. Yours is the most linear progression from beginning to end recorded that I have. So I've been using yours, which also is very convenient because Betty goes through some transitions of voice as she changes android bodies from being in a ship to being in a makeshift to being in the right android body again. That it was also made it really easy to tweak her sound, having it all in one big batch like that. So
Yeah.
yeah.
Nice.
He's nice.
It's a very nice.
But yeah, I think I'll be
6 shorts.
have time to work on that again sometime, maybe in January.
2 of the shorts are just things that I did at home.
Yeah. The neurogaming one and the thing about brains. Here we go.
And then I did like a box opening of our camera.
So duplanted memory, neurogaming, mind reading.
Ulysses wants to know what we're watching this season. What shows?
Alcohol.
We're watching alcohol.
Rebel video. Is that you opening something?
Yeah, the camera.
Ulysses, what am I watching this season? I don't know that I'm watching any of this thing this season because I only watch things on the Netflix. So everything I'm watching, everybody else has already seen just about, except for like House of Cards, Lilyhammer, and Orange is the New Black, or whatever that's called.
Yeah,
whatever Netflix produces itself, I'm pretty much watching.
I'm way behind on everything because I still have a young child.
I'm more waiting for new video games to come out than anything else. I'm waiting for the new Fallout game to come out. I'm waiting for more Sherlock.
More Sherlock, more Doctor Who. Yeah.
Yeah, I watch mostly comedy things with the short amount of time that I have. I just want to make sure that I'm laughing.
Mm-hmm.
Oh, speaking of which, had the kids. We camped in the living room last night, and starting about 6 o'clock, 7 o'clock on for the rest of the evening, through all kinds of snacking and everything else, we made it through season 1 of Alf.
Oh, awesome.
Such a good show. And I love it, the fact that there's all sorts of current event pop culture references in Alf, which of course are now like 30 years old.
Oh my gosh.
But yeah, it's a fantastic show. I guess that's the show I'm working my way through this season.
I still need
Alf.
to start watching Doctor Who, but I'm not sure where to start.
From the beginning, which
Start at the
do you have the
beginning?
Hulu?
Yeah, I have Hulu and Netflix. But
Okay,
if I just want
the
to, if I want to
Hulu-
watch, here's what I'm asking. I want to watch one episode that will
Uh-huh.
get me excited and make me then want to start from the beginning.
Ooh.
Oh, the first episode does that.
Are you sure? There's
Yeah.
No, the first episode, it's good. Like this of the new ones. The first episode of the
No,
new.
not of the
Are
new
you talking
one.
about the old ones?
You have to start the second season of the new ones, maybe.
Yeah.
But to really get the feel for it, the first episode ever is fantastic.
Yeah.
It's what's her name? The girl. She's doing super good at school, but a little too well. Like, she's complaining that the textbooks have got things a little wrong. They've got the French Revolution all wrong. She's doing extremely advanced mathematics without having reference her textbook. She's causing the interest that brings the first couple companions, who are teachers, on board with the first Doctor. It's a good episode.
Hmm.
Yeah. Oh, if you want one that just makes you realize how insanely good the show is, yeah, Blink, which is what, season 4? Is that season 4 of the new episodes? Blink. Yeah, it's the 3rd season. So okay, thank you, Gord. 3rd season. Blink is probably the masterpiece of all episodes.
Okay.
And anything with the 4th Doctor is worth watching.
So I need to be tweeted at with a specific season number and episode number, and then I'll find it. And I'll watch it. I'll do my homework. Wow, the shark video already has 478 views. That was fast.
Mm-hmm.
I have to send this to the PR department of the aquarium.
Yeah, no, absolutely.
They're going to want to see it. Oh my goodness. Kiki, it's so cool that you can edit things. I don't think I could do it. It's too hard.
It's not that hard, but it took a while, and it was yeah. I have to figure out how to use the slate on my audio recorder so that I can actually. There's like a cable that I can get that I have, but I have to figure out how to set it up correctly so that the audio in the camera syncs with the audio in the audio recorder, and so that I can slate it, so that I have like a clear point to line up the audio with the video from the audio track.
Mm-hmm.
And I did not have that, so that was fun. I spent like moving the audio around. I'm like, move it. I'm like, no, over there. Okay, trying to get it exactly right, so it didn't. So there wasn't like a mismatch between the words and the mouths.
That sounds awful.
I actually really had a lot of fun editing this. I really enjoy editing. I'm not fast, but I like it. And I don't know. I'm not the best editor, but it worked. right? Came together.
It looked good to me.
Yeah, I need to learn how to do more of the motion, like graphic-y type stuff. I don't know how to do anything related to graphics. So when it comes to like, fonts and things and shapes and putting things on top of things to move, like layering stuff, there's other things that you can do like in editing to like you put things on top. This is the part of editing that I don't know enough about at all. I'm at a loss when it comes to how to do that. But
hey, I gotta bail. Happy Turkey Day.
Happy Turkey Day, Justin.
I will see you on the other side. Remember to yell at your relatives. It's good for the kids.
What if there aren't any children?
Well, have a good time.
Maybe you should yell about. Maybe you'll get yelled at about that, Blair.
Oh, I will.
maybe that'll be the source of the yelling.
I saw the doctor today, and she goes, "Still single, huh?"
Oh my goodness.
The doctor did that too.
You're like, do you want children?
Leave me alone.
You're still single?
Yeah, I've been holding out this whole time for an unpushy doctor to marry, but still haven't met one. Yeah, still haven't met an unpushy, unnosy doctor because that's what I was looking for. That's what I had my heart set on.
Honestly, like 5 doctors in the past however many years, and all of them have said some matter of clock's ticking, sweetheart.
Wow. Seriously?
You got a boyfriend?
That's just too much.
Or you can hit him back with, yeah, I'm just waiting for a health plan that covers pregnancy. Can you work on that from the inside? Do you know anybody you could call?
Yeah. Oh, do I just need to marry the first man I meet on the street to have babies? Is that what I should be doing right now? Is that what you suggest?
Yes, okay. Blair. Yes.
Okay.
That's what you need to do. No. Absolutely do not do that.
No.
Actually, you know what? You should do that because honestly,
here comes Justin's,
no guy is ever going to be good enough for you, Blair. I truly believe this. I truly believe no man is worthy of you. Okay? But the child you have is going to be more important to you than whoever donated the sperm in the first place. That way, you won't even care at that point. That kid's going to be more important to you than anything that you've done thus far in your life.
Mm-hmm.
Well, I'm not ready to be in charge of a human life right now.
What?
I'm not ready to be in charge of a human life at this time. Thank you.
I would rather be.
What do you mean in charge of a human life? They're just kids. It's not like it's a human life. Look, you don't have to be that in charge of them in terms of their life. They are their own people. You're going to be a good parent. They're going to be around a good person growing up. This is the problem. The people who should be having kids are the ones who overthink it and be like, oh my gosh, there's so many levels of responsibility that I have to get right. Those are the people who would get it right. It's the people who are like, oh, kids, whatever. They're the ones having kids. Just you go listen to your family, listen to your doctor's advice, Blair, and go get impregnated by the first person that takes you out.
Pass. Hard pass.
Good.
Hard.
I'm telling you,
Good
I know, it's hard to be Blair, and you're trying to find somebody who meets your standards. There is no man on this earth who's going to meet this standard,
I'm
Blair.
just waiting for a man who can do a nice dance.
Right? A man with a good standard.
Solo. And then I'll be all set.
Okay, then you
Blair,
need to start taking like flamingo lessons or something, right?
Blair, did I tell you that I saw a peacock spider on Halloween? That there was a woman dressed with like a peacock spider thing, and I stopped. And I looked. I go, "Are you a peacock spider?" And she went, "I am." And then I walked away, and then I came back and I said, "Can you do its dance?" And she goes, "I can." And she did a little peacock spider dance for me with her. Yes. Just like that. It was awesome.
Wow. That's amazing.
It was amazing. I was like, oh, I can't believe I forgot to tell you that. I was like, I have to tell Blair.
I might have to do that next year.
It was pretty awesome.
Oh, that's such a great costume idea.
It was great.
Oh, man.
Peacock spider.
Ulysses, it's not that the world needs more kids. It needs more kids raised by people I like.
That is true.
It's,
It needs
it's part-
more children raised by parents who care and are going to put the resources toward their children to educate them and make them thoughtful, caring, critical members of society.
Phil Caffrey is asking, any science here today? This is the after show. The science all happened. There was an hour plus, almost an hour and a half of science.
We had a really
meetings that
long
took place
show.
Before the chit-chat rigmarole of the after show ensued. I do have to go, though. I've got a taxi cab waiting. I gotta go.
You have a
night,
taxi
minions.
cab waiting for you right now.
It's me. I'm the one. I've got to drive cab at night now to support all these damn kids. Good night.
And on that note,
Really? Are we stuck between your two jobs now?
Yeah, it's all. They're all bumping up. There's actually 3 of them now. This is just one more thing I do in a series of things that I do in order to feed all these parasites that I've brought into the world. That I love very much.
That you love very much, yes. They're fabulous. Wonderful
night, everybody.
little parasites. Have a wonderful Thanksgiving. Thank you for being a host on TWIS. Thanks for being a friend.
Aw.
Thank you. Have a good night. And Blair,
yes.
I think I should probably go too. Should we go?
Yeah.
Should we call it?
I have to bake 2 pies.
Do you still have to bake your pies?
I'll probably do it tomorrow morning, not tonight.
Yeah. I have to bake pie. I have to bake pies. I have like 2 Thanksgivings going on. There's a lot of
Thanksgiving.
there's a lot of cooking happening in my life over the next few days. I think I just need to lie down right now.
Good call.
I need to find a new book to read. That's what I need to do. Yeah.
What kind of book? Like a nonfiction heady book, like pop?
I've got plenty. I've got plenty of nonfiction heady books.
Well, if you want something popcorny and fun, I have a suggestion.
Yes.
Outlander.
Outlander. I've never read that.
You should read it. Try it. Don't expect fine literature. Expect a good time.
A good time. Wonderful. It'll keep me busy.
It is historical fiction mixed with science fiction, mixed with a little sprinkle of romance novel.
Nice.
It's really good.
I like that. That sounds fantastic, and it sounds like it could continue to keep my husband off my back about the werewolf and vampire books. Yeah.
And it's Scottish. It takes place in Scotland, and it was written.
So it's not crap.
Exactly. And it was written by a woman who got a PhD in zoology and ecology. And one day turned around and was like, I'm going to write a book. And then wrote 8.
8.
I think it's 8 in this series.
Wow.
Yeah.
Awesome.
It has nothing to do with ecology or zoology. It has a little bit of
these books.
It has a sciencey twist sometimes. Like the woman was a nurse, and so she knows how to heal people and stuff. And so there's like sciencey-ish stuff. I don't know. It's pretty cool.
Mm-hmm. I will have to check it out. I'm typing. Fonzo Blessed 3 says, love y'all. Y'all rock. Thank you. Thank you. Ben Rothig, go make something foodish for dinner tomorrow. And Phil Claffey, good night. Thank you. Thank you, everybody. Ulysses is going to be drinking hard cider tomorrow, of course. What else is going on? Thank you, Ulysses. Thank you, Hot Rod. Gourd. Pie. We want to eat pie. Rights for robots. Yeah. All right, everybody. Who else is there? Everybody in the chat room. Ben, Gord, Identity 4, Beef, B Tree, CR1, Darkcon, Eddie P, Flying Out, Fonzo Bless 3, Hot Rod, Huff 3er, Huffer, Iowa Minion, Janice Goo, LPT1, Normod, Panda Squirrel, Phil Claffey, Protonic, Cortinamus, Cortonanonymous, Rights for Robots, Rorks, Cymink, 618, Sigfault, 618, are you the same people? SMB and FLA, Tabitha's Habitat, Twit Refugee, Ulysses, Web21, Whiskey Renegade. Thank you! Thanks for being in the chat room. Thanks for hanging out for the after show. Thanks for hanging out during the show and just for being here. It's Thanksgiving time, right? I am full of gratitude for all of you. And Blair, thank you so much for being a co-host.
Of course, I am so happy to be here.
I'm glad you are part of the show, so thanks a lot.
I'm so thankful for TWIS.
Me too. I really am.
Really changed my life.
Awesome. Awesome. Good.
All right.
All right, everybody. Have a wonderful Turkey Day if you're here in the United States. I hope you have much round, full bellies and much to be thankful for. And may your cups runneth over. And hopefully we'll see you again next week for more This Week in Science. Thank you. I'm going to stop this broadcast now because I just can't say thank you anymore. Just thank you. Thanks a lot, everyone. Good night.
Bye.